I'm Janis Klovins. I'm a professor at the Latvian University and head of the Scientific Council at Latvian Biomedical Research and Study Center. These are official positions, but I'm called scientist because I have my own research group at this institute where I perform some research on the human genomics and microbiome studies in human populations. One of the biggest aims in Latvia right now is the establishment of the human genome reference for the Latvian population, and that very well coincides with the many cohorts that we've been establishing in the past within the National Biobank of Latvia. So this is both the population-based and disease cohorts that we're trying to link together the different and to find biomarkers that are coming out from different omics. So my own research interest mainly relates to diabetes and other metabolic disorders, but I've been actively involved also in COVID research in the recent past, and we're also still investigating many COVID complications with respect to different really complications of neurological and renal cases, so that we're trying to find biomarkers, how to predict them and how to really treat them. Indeed, when we first contacted we were contacted by MGI to try this really interesting new technology. So actually that changed the way how we really perform our research, because that was for us quite a difficult time to really find a good and reliable sequencing technology. So basically immediately started to use for all kind of purposes. So the first instrument that we had was a G400 that was used even for the whole human genome sequencing because we didn't have that ability with the previous instruments, and we very often use it for the metagenome, also for RNA-seq. So there was all kind of different application, and of course for the COVID, because at least for 2 years we were the main center in Latvia that provided the SARS-CoV-2 sequencing and then reported results back to the disease and control center. So basically that was the main instrument that we were using all over the years. But then with this establishment of a genome reference project, of course we needed larger instrument, and then we agreed with the MGI to use this T7 that is now extensively used for the whole human genome sequence, and of course then other automates that help to produce the libraries. This is also very good instruments from the MGI to use. Of course, we're looking forward towards genome medicine. So basically, this is something, and then not only the human genome, but also linking it together with all other kind of omics. So obviously, we really what we hope that we can kind of stop only doing scientific research, but also trying to introduce the knowledge that we have created into the clinics. And there are some defined project that we already doing that, like in the monogenic diseases, like in the familial hypercholesterolemia, that we already using this technology both for the monogenic disease and also for the polygenic risk score calculation. So, and this is something that at least my future goals actually are two. So first of all, try to involve more and more people to also do the kind of this social science that we directly are involving people, reporting results back. So working in the area of this predictive medicine. So and then try to find the cause before the disease actually has happened. So this is one direction, and then another one is really move forward to implement the knowledge that we created into the clinical settings.